Hi there, I'm Professor Patrick Degener and this is bioelectronics. So in this lecture we're going to cover the concept of neural computation, what type of computations neurons can perform, and how do neurons do the adding, subtraction and memory functions that achieve these computations. So first of all, let's consider the concept of computation in terms of a silicon system that exists in mobiles and computers and laptops, etc., etc. So typically, some information will come into your central processing unit, and this typically is in the form of an instruction to perform a particular computation and perhaps a memory address. The processor grabs the required or fetches the required memory element, takes it back to the process unit, and then that instruction tells what's called the arithmetic logic unit to perform the required calculation, and then out pops the answer out the other side. And this could be a command to to control a hard disk, or it could be just another memory location. But if we focus specifically on the computations themselves, you can think of the computation as something like this, where you've got y equals ax1 plus bx2 squared, where x1 and x2 are inputs and a and b are settings. So it's not just performing a calculation on the two input values, but you also need some kind of setting values, and this effectively is memory, and offsets. Now it's worth noting that the values in the digital world are stored as either a 0 or 1, and a sequence of these, 1 0 1 0 or whatever, in 8 bits or 16 bit or 64-bit form. When you come to neurons, information is transmitted in an action potential, which is digital in amplitude, but is not digital, or it is analog in time. So it's still a kind of an analog form of information, unlike the digital world. It's also notable that prior to the formation of an action potential, neurons acquire signals, which I'll explain in a moment, and those values are analog values. So computation in computing systems and neural systems are quite different. And if we think about neurons, neurons aren't processors. They're much simpler structures than processors, but they're much more complex than individual transistors. So they're somewhere in between, in terms of capability and in terms of what they do. So if you take a simple neuron, such as this one, a neuron will take inputs from sensors or from other neuron cells. It will perform some kind of processing function, and typically this is a summation function. But in order to perform the process properly, it needs some memory. And as we described before, where you've got, y is equal to ax1 plus bx, maybe squared, x2, etc., etc., each one of those inputs has to have a certain weighting, which could be considered a memory. And then once a decision is made to form or not to form an action potential, or a sequence of action potentials, the information is then transmitted, where it then goes to perhaps another neuron, or it goes to a muscle, or perhaps some component in the peripheral nervous system. It could be a heart, cardiac myocyte, or it could be the pancreas or something else. Now, how the neuron performs this is described in this diagram in the center, where you have a central neuron, and then this central neuron is synapted by 4 neurons, so neuron 1, neuron 2, neuron 3, and neuron 4. And each of these neurons has some signal, A1, A2, A3, A4, and they have a synapse with a particular synaptic strength, S1, S2, S3, and S4 respectively. And then the neuron in the center, takes these signals coming in and adds them all up. And conceptually, you can see that in the graph here to the top left, where, for example, the A2, the red one, could be inhibitory, so that's a negative, and the others are positive. And then when they all add up, maybe, in this particular case, the summation could go beyond the threshold, this black line, and if it does so, then this will activate an action potential, which then traverses down the axon down to the next target. So we can think of the neuron then as performing a function which is a bit like this, A1 S1 plus A2 S2 plus A3 S3 ... and you could have hundreds or even thousands of inputs to any particular cell. And if we take our model of the neuron, which is that the voltage, that the voltage in a neuron is given by the total amount of charge coming into that neuron divided by the capacitance. This, in a sense, is that this charge is the summation of the dendrites. That should be one. The summation of the dendrites divided by the capacitance. Well, the summation is just the strength of the signal times the strength of the synapse, and then you sum over all of these particular synapses. So it's a very straightforward concept. It's just a summation function. But the real trick is in the weightings of these particular synapses. That determines whether one type of neuron has a more important input than another type of neuron. Now, in order to understand these processes, it's very important to think about the molecular forms. And there are two components in the post-synapse, and these are two types of receptors. The first is called the ionotropic receptor. And in an ionotropic receptor, this is basically just like your standard ion channel. It's a standard channel which opens up, closes, except that it's activated chemically by neurotransmitters. And if we want to have a conceptual equation for this, we can say, well, okay, we have your standard Hodgkin-Huxley type equation where there's a conductance, G, times your, the voltage difference across the membrane between the Nernst potential for that particular ion channel and the overall voltage. And then you can think of the fact that the ion channel is open for a certain period of time, because the integral of current will give you charge. Now, the other type of synapse is the metatropic receptor. And in the metatropic receptor, you've got a subtle difference in how this works. So first of all, the receptor has this receptor here has no ion channel beside it. It's not directly connected to an ion channel. Instead, the receptor activates something called a G protein. And the G protein effectively creates a cascade of chemical activity where it starts to break up a particular molecule, or will typically break up a particular molecule, which in turn allows for the activation of an ion channel related to that particular chemical. So you can have positive and negative. And this allows us, in a sense, that a single receptor can cause an amplification where lots of ion channels are then activated by the single neurotransmitter. It also allows for a reversal of activity to happen, where you can the amplifier could actually just simply have a value of -1. But we'll talk about this a little bit later in the vision section. So the concept model in this case Well, it's your, it's your conductance times the voltage difference again. This is your, Hodgkin-Huxley type equation, and you've got your time, except that the time constants can be a little bit different here. So these things are slower and take a bit longer. But now you've got your amplification, whatever that amplification factor is. So it's somewhat different to the ionotropic case. It's slower, but you can have a much greater sensitivity. And the light sensors in our eyes operate on this principle so that individual photons can effectively be seen by the human eye, which is quite incredible. Now, we mentioned before about the synaptic cleft and roughly speaking how the synapse worked, and we mentioned that as as neurotransmitters are released, they have to be removed again, and there's really three possibilities for that removal process. The first is just that it leaves and goes somewhere else and spreads out. The second, which is really, really important, are the enzymes which come in and break them up. So you could have enzymes that break up glutamate, or enzymes which break up acetylcholine into the component parts of choline. And then the third process is what's called reuptake, where the presynapse just sucks these chemicals back again. So that this is a very efficient form of taking the chemicals back so that you can use them again the next time. And this is where we have to talk about cellular memory and potentiation. And we kind of discussed this briefly before, but just to emphasize this again. So there are two types of cellular memory, and these relate to the efficiency between the presynapse and the postsynapse. So if you've got a high efficiency of information transfer, it's potentiated. If you've got a low efficiency, it's depressed. And note that this term depression has nothing to do with the psychology term depression, which is about whole brain activity. In this case, it's just a reduced transmission between the presynapse and the postsynapse. And this potentiation and depression can be short-term or it could be long-term. We can have long-term memories, or we can have short-term adaptations. As we're walking down the street, we may find that the terrain is a little bit rougher and we may need to adapt our walking form, ice versus gravel. So these are short-term changes. And we need positive potentiation and also negative depression. So don't think of these as good and bad. We need both of these in order to perform the computations and keep our brain modulated in the correct form. So how are these memory tasks performed? So there's potentiation, where there's a lot of information transfer, and there's depression, where there's a limited information transfer. Well, first of all, let's look at this image. Sorry, it's poor quality, but it's the best I could get from this Nature Reviews paper. So let's look at the presynapse. So as an action potential comes down, because the nature of an action potential, it's opening up ion channels in order to, in order for the action potential to traverse. It's not just sodium and sodium that enters the cell and potassium that exits the cell, but you also have an influx of calcium. And we've talked about this before in the Nernst potential and the Hodgkin-Huxley equation. And calcium has a relatively limited effect on the overall action potential, but it's very, very important when it comes to the adaptation of cells. So what the calcium does once it enters the cell is it starts to activate a chain reaction of chemistry, and that chain reaction increases the number of vesicles that are created and eventually get released to the postsynapse. So before, when you have a depressed synapse, only maybe one vesicle gets released, and very few neurotransmitters get released into the synaptic vesicle. But now, as more action potentials come through, this starts to create a process where now there's more vesicles and more neurotransmitters being released, which can then activate the post cell. Once this happens, as you start having neurotransmitters activating individual receptors in the post synapse, again, it's not just sodium which enters the cell. You now also have calcium entering the cell. The calcium starts to activate processes which start to produce more receptors. If there are more receptors in the post synapse, when they get activated by the neurotransmitters, more charge enters the cell, which then has a greater impetus for the neuron to integrate that and create an action potential. Now, last but not least, it's worth mentioning drugs. Don't do drugs, kids. But in all seriousness, in the computation world, we sometimes think of all these synapses acting alone by themselves, but there is an effect from broad active drugs. One interesting aspect are some of these nerve agents like sarin and VX, and what they do is they destroy or inactivate the acetylcholinesterase enzyme, which eats up all of the acetylcholine that our motor neurons transmit. And that means that in the synapses of between our motor neurons and our muscles, there's just this constant signal going through, and the muscles spasm, and eventually your heart stops and you die. And the other side of things is that, of course, if you have drugs which activate dopamine and serotonin systems, they can affect your mood. The neurons which are in the areas of your brain that affect your mood and your emotions and your feeling, these can then be activated. And if we go away from the illicit drugs, you can think of serotonin reuptake inhibitors, which are the seroxatats and so forth. These are the core drugs which are used to treat clinical depression. This is the big psychology depression, not the little synaptic depression. And these have quite a strong effect by basically preventing the reuptake of serotonin in the synapses in these regions of the brain. So hopefully you found that interesting. Thank you very much for listening, and see you at the next lecture.